Yes, you're good.
Perfect. Hello, everyone. Thank you all for coming to watch our presentation. My name is Ramtin Ghafoori. I'm a third-year biology major, and I'm working at Dr. Michael Adams' lab. My research is about how ecdysis triggering hormone, or ETH, influence courtship behavior on male Drosophila. So let's just start by talking about ETH. Ecdysis triggering hormone, or ETH, is a peptide hormone that's released from endocrine Inca cells. These two figures show the Inca cells, the green dots, green fluorescent or Inca cells. The top one are in a larva, and the bottom one is in adult Drosophila. The non-function of ETH is to trigger ecdysis behavior, which is cuticle shedding during juvenile development, and also it influences the reproductive physiology and behavior in adults. Based on past studies on fruit fly behavior, male fly is not engaged in mating after they mate the female, and we believe that this concept is related to impact of ETH. So my research hypothesis is that ecdysis triggering hormone inhibit post-mating courtship behavior in male fruit flies. These two figures are from past studies done in Dr. Adams' lab. The left figure shows ETH deficiency increases courtship behavior, which they the Inca cells were ablated in flies, and since there is no Inca cells, there is no expression of ETH and no hormone of ETH is produced. So the flies are ETH deficient, and the courtship index, which is basically index telling us whether male pursues, whether male try to mate the female or not, is much higher than the wild type. The figure to the right also tells us that ETH injection reduces courtship behavior, which we believe that although this study seems true, we think that the data we got from this study is not very accurate because the amount of ETH injected was much higher than what could be produced by endogenous system. So my research is kind of similar to the past, but it's different in a way that we are trying to release endogenous ETH, stimulating release of ETH naturally from Inca cells, and see how ETH influence the courtship behavior. So in this study, we use channel rhodopsin. Channel rhodopsin or light-gated ion channel, which activates by red light. And as soon as red light penetrate the insect cuticle hitting the channel rhodopsin, it opens and sodium and calcium enters the cell, causing the cell to depolarize. However, calcium entry to cell to the Inca cells causes ETH to leave the Inca cell via vesicular exocytosis. It leaves to the hemolymph and it can starts functioning. Naturally, in wild type, we don't have any channel rhodopsin on the Inca cells. So the first step is to cross Ghafoor ETH line with UAS channel rhodopsin line to produce the Inca cell specific expression of light activated ion channel. The way that Ghafoor and UAS system works is that Ghafoor is a driver gene. So when we cross these two lines, the progeny has, as soon as the precursor for ETH hormone gets expressed, it activates the expression of Ghafoor. Also, Ghafoor is a transcription factor, so it starts activating UAS, and the gene of interest starts to getting expressed, which in our case is channel rhodopsin. After getting our flies, it takes about basically after 10 days to 2 weeks, we separate the pupa, fruit flies at the pupal stage, right before becoming adults. We're going to sex them and putting in the small test tubes. The reason that we are doing this is because we don't want our flies to have any social interaction with other flies. So their courtship is not impacted by interaction with other flies. They are going to close inside these small test tubes and there's no interaction with other flies. So our experiment has 3 samples. The first sample is Ghafoor line that we use. Sample 2 are UAS flies. which these two are control of our experiment. The experimental group is Ghafoori UAS that's crossed, and we are going to put all the samples, like 5 Drosophila from each sample, inside the courtship chamber, and we are going to put the courtship chamber under the red light to activate the channel rhodopsin. However, Ghafoori flies and UAS flies, they don't have expression of channel rhodopsin, so there should be no impact on their ETH level due to red light. Here's a video of us doing this experiment. We see that red light is flashing over the courtship chamber, and we've already put flies, male flies, because the courtship index is related to male flies. It's an index for measuring male mating females. After that, we are going to put, so courtship chambers has like chamber, and there is a gate inside each chamber. So we are going to put one female and one male on each side of the chamber. Gates is closed, so there is no interaction. And we are going to do this without using any anesthesia, because usually when we take care of fruit flies, we can use CO2 and we flip the flies when the flies are asleep. But in these experiments, we try to not use any CO2. Using this device, I try to use my mouth and basically put the flies in the courtship using this, because CO2 may impact their courtship behavior. And after that, we are going to put the courtship chamber under the camera, open the gates, and start recording for 10 minutes. This is the sequence of behaviors we expect from fruit flies. The male starts to following the female, then start tapping. The male starts to sing by extending their wings, and this until the copulation, which is the last step. So then we are going to watch the video measuring how many seconds male try to copulate and mate with female, recording all the times, and calculate the CPI, which is the courtship index. It's a percentage that male pursues female in 10 minutes interval. If they copulate successfully, we stop recording at that time. If not, we do not have successful copulation, we stop at 10 minutes. And this time is like we are dividing the time they try to copulate by the 10 minutes. And the expected outcome of this experiment is that rising ETH level leads to courtship inhibition of the courtship index, and the courtship index of channel rhodopsin expressing flies exposing to red lights will be lower than our control experiment. And this study shows us how hormonal state of fruit flies impact their courtship behavior and physiology. Thank you all for listening. I'd also like to thank Dr. Adams for his help and support, and I will be taking any question.